So right now, sitting just beneath your ribs, there is a pool of hydrochloric acid, and it's actually so concentrated that it could quite literally dissolve zinc or, eat right through solid wood.
Which is pretty terrifying when you really stop to think about it.
Right. Yet somehow it doesn't just digest your own body. I mean, we tend to think of the stomach as this kind of passive biological storage tank. You eat a meal, it drops down into this fleshy pouch, and it basically just, well, sits there waiting for the intestines to do the real work.
Yeah, and if you think that, you're actually in very good historical company. I mean, the idea of the stomach as a passive fermentation vat, it dates all the way back to ancient Greek medicine and Galenic humoral theory.
Wow, really? So they thought it just sat there.
Pretty much. For centuries, the prevailing wisdom was that the stomach didn't really, do anything active. It was essentially viewed as this process of controlled putrefaction, just holding the food while residual body heat and, well, time caused it to rot into a usable state.
That is, I mean, that's just so gross. But the snack of sources we're looking at today completely shatters that centuries-old myth. We've pulled from clinical gastroenterology textbooks, medical history journals, and the latest surgical research to bring you this deep dive into where digestion truly ramps up. The mission today is to pull back the curtain on an organ that is actually a highly active, neurally regulated chemical reactor, a brutal mechanical blender, and even a sophisticated hormone-producing command center. Okay, let's unpack this. Where do we even start?
Well, the reality of what is happening in your upper abdomen right now is structurally and chemically fascinating. We're going to explore the exact mechanisms of how your stomach physically and, molecularly breaks down a meal.
Right.
But to truly appreciate the physiology, we kind of have to look at how that ancient medical dogma was finally overturned. And it required a wild combination of empirical scientific genius, a close-range shotgun wound, and later on, a doctor willing to drink a literal petri dish of cultivated bacteria just to silence his critics.
It is honestly one of the wildest timelines in medical history. So to understand how we figured out the stomach wasn't just a passive pouch, we have to head back to 1822 in the Michigan Territory.
Yeah, the American frontier.
Exactly. We're at a remote army outpost with an American surgeon named William Beaumont. And one day, a young French-Canadian fur trapper named Alexis St. Martin is accidentally shot in the abdomen from just a few feet away. Beaumont treats him, fully expecting this massive tissue damage to be fatal. But miraculously, St. Martin survives.
He survives, yes, but the tissue heals in a really highly unusual way. It resulted in what is medically known as a gastric fistula.
Which is basically a hole.
Yeah, essentially the abdominal wall fused completely with the stomach wall. So it left a permanent open window directly into St. Martin's working stomach, covered only by this loose flap of tissue.
I mean, most physicians would just document that as a horrific tragedy and move on.
Right, just patch him up as best they could.
But Beaumont realizes he has been handed this once-in-a-lifetime scientific opportunity. He can look inside a living, functioning human body and watch digestion happen in real time. So over the next decade, he runs hundreds of experiments.
Hundreds of them, yeah.
Beaumont essentially becomes the first person to look inside the body's engine room while it's running, and he completely rewrites the owner's manual. I mean, he literally tied pieces of food like meat, cabbage, bread to a silk thread, and then dangled them through the fistula into St. Martin's stomach.
And then just timed how long it took for them to dissolve, which is honestly astonishing empirical science for the 1820s.
It's so audacious, just dangling meat on a string into a guy's stomach.
It really is. But Beaumont went even further. He extracted the gastric juices directly from the fistula into glass vials, kept them at body temperature using sand baths, and placed food directly into the vials.
Oh, wow. So he replicated the stomach outside the body.
Exactly. And by doing this, he proved definitively that gastric juice was an active chemical solvent, because the food in the vials dissolved at roughly the same rate as the food in the stomach. He proved digestion was driven by highly specific chemistry, completely independent of the stomach's physical movement.
So the food wasn't just rotting.
Right, it didn't putrefy. It was actively broken down by a solvent.
And that discovery opened the floodgates. Once Beaumont proved there was a chemical solvent at work, the race was on to figure out what that solvent actually was. By 1823, an English chemist named William Prout identified the presence of hydrochloric acid in those gastric secretions.
Which was shocking at the time. The idea that a living organism could manufacture a strong mineral acid like the kind of corrosive liquid used in heavy metallurgy, that was highly controversial.
Yeah, they must have thought he was crazy. And then, in 1836, a German physiologist named Theodor Schwann isolated an enzyme in the stomach, which he named pepsin, proving that the acid wasn't acting alone, right?
Right. It had a biological catalyst working alongside it to specifically dismantle complex proteins.
Here's where it gets really interesting. We moved very quickly from the fermentation vat model to this chemical reactor model. But it takes a tag team effort of brute mechanical force and extreme chemistry to turn a solid meal, like a thick steak or a fibrous apple, into usable fuel.
Yeah, you need both. Let's look at the mechanical breakdown first. Throughout most of the gastrointestinal tract, the muscular walls are composed of just two layers of smooth muscle, a longitudinal layer and a circular layer.
Okay, 2 layers.
Right, and these allow for peristalsis, which is the wave-like squeezing that just propels contents forward.
But the stomach operates under totally different structural rules. It actually has a third muscle layer, the oblique layer.
Right.
If you just had the standard 2 layers, the stomach would merely push the food forward.
Exactly.
But that third oblique layer, it functions kind of like the agitator in an industrial washing machine. It allows the stomach to actively churn, twist, and pulverize the food.
Yeah.
It's not just moving your lunch. It is violently beating it up.
It is an incredibly turbulent environment in there. Roughly 3 times per minute, initiated by these pacemaker cells in the stomach wall, a really powerful wave of contractions sweeps down from the upper stomach.
Just smashing everything.
Literally. It mixes the food with the gastric juices and forcefully smashes it against the lower part of the stomach, which is called the antrum. And the antrum actually has thicker musculature specifically designed to grind this material against the closed exit valve.
But, brute force alone just leaves you with a bruised piece of meat. To actually dismantle the food at a molecular level, you need that specialized solvent. If you look closely at the mucosal lining of the stomach, it's heavily textured with these microscopic wells called gastric pits, which lead down to gastric glands. And these glands are packed with highly specialized cells. You have the parietal cells, which are basically these microscopic acid factories flooding the stomach with hydrochloric acid.
And the mechanism behind those parietal cells is just a marvel of cellular biology. They contain proton pumps, specifically the HK-ATPase pumps.
Okay, proton pumps.
Yeah, and these pumps use massive amounts of cellular energy to actively force hydrogen ions out of the cell and into the stomach cavity against a staggering concentration gradient. They're literally pumping protons against a gradient of 1,000,000 to 1.
1,000,000 to 1? That's insane.
It is. This active transport is what drops the pH of the stomach down to a highly caustic range of 1.5 to 3.5.
And right alongside those parietal cells, you have the chief cells, which secrete a substance called pepsinogen. And what's fascinating about pepsinogen is that it is secreted in an inactive state, like a proenzyme.
Right, because if the chief cells secreted active pepsin, it would destroy the very cell that created it.
Exactly, it would just eat itself. Instead, pepsinogen is released, and it physically requires that highly acidic environment. When the proenzyme hits the hydrochloric acid, the low pH actually causes the protein to unfold. The acid cleaves off a specific masking sequence of amino acids, instantly converting it into active pepsin.
And pepsin is basically a set of biological scissors designed to hunt down and snip long protein chains into shorter, manageable peptides.
Which brings us to the most glaring paradox of stomach anatomy. We have a muscular sac violently agitating a pool of acid strong enough to dissolve zinc, filled with an enzyme specifically designed to rip proteins apart. So if it's doing all that, why doesn't the stomach, which is constructed entirely of protein-rich tissue, just digest itself?
Yeah, it's a great question. The protective mechanisms have to be completely flawless. The stomach is locked in this constant, perfectly balanced arms race between optimal digestion and total self-destruction. And it protects itself through 2 primary mechanisms.
Okay, what's the first one?
First, the epithelial cells secrete a specialized viscous mucus bicarbonate barrier. The mucus physically coats the inner lining, acting as this thick gel-like shield, while the bicarbonate ions get trapped within that mucus layer to chemically neutralize any acid that manages to seep through.
So it's like an alkaline force field.
Pretty much. It keeps the actual surface of the stomach lining at a safe, near-neutral pH.
And what's the second mechanism?
The second mechanism is sheer regenerative speed. Because despite that mucus bicarbonate shield, the caustic environment does cause constant microscopic damage to the tissue. To counter this, the stomach relies on highly active stem cells located in the neck of the gastric glands.
So they're just constantly rebuilding.
Exactly. These cells continuously divide and migrate upward, meaning the stomach lining completely replaces its surface epithelial cells every 3 to 5 days.
Every 3 to 5 days, that's unbelievably fast.
It is essentially repaving the highway while the traffic is actively driving over it. If either of those mechanisms, the chemical shield or the rapid cell turnover, fails, the acid reaches the vulnerable underlying tissue, and that results in a peptic ulcer.
Wow. What stands out when you look at all this extreme mechanical and chemical violence is that the stomach's primary digestive portfolio is actually quite narrow. Its main job is dismantling proteins and physically liquefying the food. Contrary to the popular belief that the stomach absorbs all the nutrients from a meal, almost all nutrient and caloric absorption happens downstream in the small intestine. The stomach is really just the demolition crew prepping this site.
What's fascinating here is that if the stomach simply dumped that highly acidic, violently churning mixture directly into the delicate small intestines all at once, it would be disastrous.
Yeah, it would just burn right through it.
Yeah, it would cause severe chemical burns and osmotic shock. The process requires precise, controlled release. So at the very base of the stomach, where it meets the duodenum, sits a thick, muscular ring called the pyloric sphincter.
And the pyloric sphincter, it acts like an incredibly strict bouncer at an exclusive club. It functions as this highly calibrated sieve, only allowing tiny particles under 2 millimeters in diameter in liquid to pass through. At this stage, your meal isn't even food anymore. It has been transformed into this highly acidic, liquefied slurry called chyme. If a chunk of food hits the sphincter and is larger than 2 millimeters, the sphincter just forcefully bounces it back into the antrum for further mechanical grinding and chemical exposure.
Right. It ensures the duodenum only receives the exact volume and consistency of chyme it can safely neutralize and process. And the duration of this emptying process depends heavily on the macronutrient composition of the meal you just ate.
So different foods take different amounts of time.
Exactly. A normal stomach will clear a standard solid meal in roughly 2 to 4 hours, whereas clear liquids, they can transit in under an hour. But the communication between the intestine and the stomach is bidirectional.
Meaning they talk to each other.
Yeah. When you consume a heavy, fat-rich meal, the moment those complex lipids reach the duodenum, they trigger the release of specific hormones, like cholecystokinin or CCK. And these hormones travel through the bloodstream right back to the stomach and actively inhibit gastric motility.
Oh, wow. So that is the exact physiological mechanism behind why a heavy, greasy meal feels like it just sits in your stomach like a brick. Your small intestine is literally detecting the high-fat content and chemically signaling the stomach to pump the brakes.
Right. It delays emptying because complex fats take significantly longer to emulsify and absorb downstream.
That feedback loop highlights a really crucial biological reality. The stomach does not operate in isolation. It is under profound neural and endocrine control. I mean, we all know the story of Ivan Pavlov in classical conditioning with the drooling dogs, right?
Sure.
But Pavlov actually won the 1904 Nobel Prize for proving the cephalic phase of digestion. He demonstrated that gastric secretion is under direct neural control via the vagus nerve. The stomach begins revving its metabolic engines, firing up those parietal cells to secrete acid, simply based on the sight, smell, or even just the psychological anticipation of food before a single bite even crosses your lips. The brain actively pre-programs the stomach.
And the stomach constantly talks back to the brain too, because it is a highly active endocrine organ. It secretes hormones directly into the bloodstream to regulate systemic functions. For example, specific G cells in the stomach secrete gastrin, which is a hormone that stimulates the parietal cells to ramp up acid production when protein peptides are detected.
Right.
But even more critically, the stomach produces ghrelin, often referred to as the hunger hormone, which was only discovered relatively recently in 1999.
Which is wild that we were still discovering major hormones in 1999. So ghrelin is produced primarily by specialized cells in the fundus, the upper portion of the stomach. And when the stomach is empty, these cells synthesize and release ghrelin into the bloodstream. It crosses the blood-brain barrier, binds to receptors in the hypothalamus, and generates the neurological sensation of hunger and the drive to forage. Then when you eat and the stomach physically distends, mechanoreceptors detect the stretching, and ghrelin production just plummets, signaling satiety.
And this perfectly debunks one of the most persistent diet myths out there. You constantly hear people say that if you just eat small portions for a few weeks, your stomach physically shrinks, making you less hungry.
Right, I've heard that a million times.
But the stomach is a highly elastic organ, much like a balloon. It stretches to accommodate a volume of food and then recoils to its baseline resting size once it's empty. You cannot permanently shrink the physical capacity of the organ just by undereating.
So what's actually happening when people feel less hungry?
The reason people actually feel less hungry after adapting to smaller portions is entirely due to neurological and hormonal adaptations. The vagal mechanoreceptors in the stomach wall reset their baseline sensitivity to stretching, and simultaneously the brain downregulates its ghrelin receptors.
So the brain just adjusts to a new chemical baseline.
Exactly. It proves that the stomach operates as a highly sophisticated signaling command center, constantly updating the central nervous system on our nutritional and metabolic status.
It is just a brilliantly orchestrated balance of neural signals, hormones, mucus, and acid. But when that delicate balance fails, things go terribly wrong. And for decades, the global medical establishment was fundamentally incorrect about why those failures occurred. For almost the entire 20th century, the dominant medical dogma regarding peptic ulcers was the stress and acid paradigm.
Yeah, the medical mantra, which had been entrenched since 1910, was literally no acid, no ulcer. The absolute consensus was that ulcers were a lifestyle disease caused by excess stomach acid triggered by chronic stress, spicy diets, and anxiety.
So what did they do for the patients?
Well, patients suffering from agonizing bleeding ulcers were just prescribed bland diets, told to reduce their stress, and given basic antacids. The underlying cause wasn't being treated because the underlying cause wasn't stress.
The way we finally discovered the true root cause is absolute stuff of medical legend. In 1982, two Australian physicians, Barry Marshall and Robin Warren, were analyzing biopsies from the stomachs of ulcer patients. And they repeatedly identified a specific spiral-shaped bacteria, which was later named Helicobacter pylori. They hypothesized that this bacteria was the actual pathogen causing the localized inflammation and ulceration.
And the medical community's reaction was intense skepticism, bordering on outright ridicule. Because, as we established earlier, the stomach is a violently acidic environment that can dissolve heavy metals.
Right, they thought nothing could live in there.
Exactly. The absolute assumption was that the stomach was a sterile environment, that no living organism could possibly survive, let alone colonize that acid bath. Journals rejected their research. The idea ran counter to everything gastroenterology fundamentally believed.
So they were just getting totally shut down.
Yeah. So Barry Marshall decided he needed incontrovertible proof.
And in 1984, in an act of staggering scientific audacity, Barry Marshall literally drank a beaker filled with a cloudy, murky broth. It was a highly concentrated petri dish culture of H. pylori that he had intentionally grown in his lab. He willingly drank the bacteria.
He did. And the mechanism by which H. pylori survives is fascinating. It secretes an enzyme called urease, which converts urea present in the stomach into ammonia.
Okay, ammonia.
Right, and this ammonia creates a localized neutralizing alkaline bubble around the bacteria, protecting it from the hydrochloric acid and allowing it to safely burrow into the protective mucus layer.
That is incredibly clever for a bacteria. So what happened to Marshall?
Within days of drinking the broth, Marshall developed severe acute gastritis, nausea, vomiting, and extreme inflammation. He underwent an endoscopy and a biopsy, which proved the bacteria had successfully colonized his stomach and was actively causing the tissue damage.
Wow.
Crucially, he then cured his own lab-induced infection using a standard course of antibiotics.
He intentionally gave himself the disease, proved the mechanical cause, and then demonstrated the cure. It took about a decade for mainstream medicine to fully accept the paradigm shift, but Marshall and Warren completely revolutionized gastroenterology. They were even awarded the 2005 Nobel Prize in Physiology or Medicine.
It's amazing.
Today, peptic ulcer treatment isn't about managing workplace stress. It's about the targeted antibiotic eradication of an invasive bacteria.
If we connect this to the bigger picture, it fundamentally rewrote our understanding of the human microbiome, proving the stomach is not a sterile wasteland, but an environment with its own specialized microbial residence. Managing that specific bacteria is critical, as the World Health Organization now classifies H. pylori as a Group 1 carcinogen linked directly to the development of gastric cancer.
This entire journey, from Beaumont pulling vials of gastric juice out of a fistula to Marshall drinking a beaker of cultivated bacteria, it intersects directly with how modern medicine manages human health today. Consider how ubiquitous proton pump inhibitors or PPIs like omeprazole are globally.
Oh, they're everywhere.
Yeah. These drugs don't just neutralize acid like old-school calcium tablets. They work at the molecular level. They cross the cell membrane and covalently bind to those specific HK-ATPase pumps on the parietal cells, physically shutting down the microscopic acid factories.
And we see this physiological knowledge applied in modern bariatric surgery as well. Take the sleeve gastrectomy, which surgically removes roughly 75 to 80% of the stomach.
Right, for weight loss.
Yeah. For years, the assumption was that it worked purely through mechanical restriction, creating a smaller anatomical pouch so the patient physically cannot consume as many calories. But the clinical research shows it is profoundly metabolic.
Because of the hormones.
Exactly. By surgically excising the fundus, surgeons are removing the body's primary ghrelin-producing tissue. The surgery alters the entire gut-brain endocrine loop. It fundamentally changes how the hypothalamus perceives hunger, which is why it is so remarkably effective not just for weight loss, but for rapid remission of metabolic diseases like type 2 diabetes.
So what does this all mean? The stomach is a marvel of evolutionary engineering. It relies on a high-energy cellular pump to create an extreme chemical solvent, pairs it with brute mechanical force from an oblique muscle layer, and turns solid food into a highly regulated slurry. It only survives this violent, self-destructive process by regenerating its entire epithelial lining every few days. Its behavior is tightly governed by the brain via the vagus nerve and driven by complex hormonal feedback loops like ghrelin and CCK. And for a long time, we were completely wrong about it, blaming stress for tissue damage that was actually caused by a remarkably adaptive ammonia-producing bacteria.
It's quite a resume for one organ. But before we wrap up, we really cannot ignore the direct hematological link that perfectly illustrates how deeply integrated the stomach is with the rest of the body. We discussed the parietal cells at length regarding their role in pumping protons to create hydrochloric acid.
Right, the acid factories.
However, those exact same cells also manufacture and secrete a specialized glycoprotein known as intrinsic factor.
Oh, and without intrinsic factor, the entire circulatory system is compromised. Because intrinsic factor binds specifically to vitamin B12 from the food you eat.
Exactly.
Without that glycoprotein binding to it, your terminal ileum, which is the final section of the small intestine, is completely incapable of absorbing vitamin B12 into the bloodstream. And without B12, your bone marrow cannot produce mature, healthy red blood cells, leading to a severe, potentially fatal condition known as pernicious anemia.
You could consume a diet perfectly optimized with iron and B12 supplements, but if your stomach lining is severely damaged, or if you've had a total gastrectomy, your blood health will inevitably crash without synthetic intervention. It proves definitively that the stomach's function extends far beyond digestion. It is biochemically essential for the vitality of your blood.
So the stomach is literally ensuring our circulatory system functions.
It is, and this raises an important question regarding how we classify this organ. We know the gastrointestinal tract is governed by the enteric nervous system, this incredibly complex network of over 100 million neurons, often referred to as our second brain.
Right.
We know that the stomach's baseline motility and acid secretion do not completely shut down when we lose consciousness, which is why consuming a heavy meal right before bed often results in nighttime acid reflux. The chemical reactor is still churning while you sleep.
That makes sense.
And we know that physically altering the anatomy of the stomach fundamentally changes how the brain regulates systemic metabolism and insulin resistance. So, if this organ controls our neurochemical drive to eat, directly regulates our metabolic set points, and operates its own independent neural network around the clock, is it possible that we need to stop viewing the stomach merely as the first mechanical step of digestion and start recognizing it as the primary sensory organ for our enteric nervous system?
I love that perspective, a primary sensory organ for the gut's brain. Well, thank you for joining us as we explored the incredible anatomy and physiology of where digestion truly ramps up.
My pleasure.
The next time you feel that physical rumble or you notice a heavy meal delaying your gastric emptying, I hope you have a profound new appreciation for the violent, perfectly balanced neuroendocrine chemical reactor churning away just beneath your ribs. Keep wondering, keep learning, and we'll catch you on the next deep dive.